Okay, so welcome everyone to yet another NIRx webinar. It really is our pleasure today to have Onn Wah Lee with us from the Bionics Institute and the University of Melbourne in Australia. We'll be talking to us about the use of heart rate responses extracted from functional neuropet spectroscopy data as a measure of speech discrimination ability in sleeping infants. So if this is your first time joining one of our webinars, of course, welcome, and we'll tell you a little bit about our company. So for the last 20 years or so, NIRx has been designing and manufacturing innovative and user-friendly and powerful NIR solutions. We focus on providing researchers with training and outstanding support and a collaborative resource environment. And of course, these webinar series are just an example of our efforts. Of course, our fNIRS solutions are developed and invented in Berlin, Germany, and they're distributed worldwide. And they are supported by an international team of scientists. So if you'd like to learn more about our company, feel free to connect with us on various social media platforms. And of course, you could email us at consulting@nirx.net if you'd like to learn more about our systems. You can also subscribe to our newsletter where you'll essentially will email you the latest product features and releases, and you'll get to hear about upcoming webinars and all sort of things like that. And we promise not to bombard you with emails, so we'll only email you about 3 or 4 times a year with just some of the exciting news and research. So for today, I'll be moderating the webinar with Eric Nelson, who is a technical consultant with us, and he'll be making sure that everything runs very smoothly behind the scenes. And a few housekeeping points. Everybody on this call will be muted. Of course, questions are welcome at any time. Please use the Zoom chat function. We will collect all the questions and ask them to Onn Wah at the end of the webinar. If you would like to, instead of typing your question, you would like to rather ask your question directly, you can always use the raise hand function and we'll call on you and unmute you so you can ask your question. This content will be available on our website. So this webinar is being recorded and will be made available in the upcoming days. And if you have any questions, you can email us at consulting@nirx.net, or if you're a current user, you could email us at support@nirx.net. Okay, so without further ado, I would like to welcome Onn Wah Lee. So Onn Wah is a PhD student, and he's investigating the use of novel imaging techniques such as fNIRS to measure speech discrimination in sleeping infants. His studies conducted at the Bionics Institute and the University of Melbourne in Australia, under the supervision of Professor Colette McKay and Dr. Wunderlich. He's an audiologist by training with a master's in audiology with clinical practice awarded by the University of College London in 2016, and a bachelor's in audiology with honors from the National University of Malaysia in 2013. Currently, he is writing his PhD thesis as well as working on publishing a groundbreaking study that identifies independent components of fNIRS speech detection and discrimination response. And after his PhD, he strives to continue developing fNIRS as a clinical tool to measure speech perception ability in infants. I hope everybody enjoys this webinar, and Onn Wah, we're very much looking forward to your talk. And you can go ahead and share your screen whenever you're ready.
Okay. All right, can everybody see my screen? Yep,
looks great.
Right, great. Okay, thank you. Hello, everyone. I'm Onn Wah from the Bionics Institute. Thank you for joining the webinar, and a special thank you to NIRx for having me to deliver the webinar today. So as Andrew mentioned earlier, I'm a PhD student who is under the supervision of Professor Colette McKay and Dr. Wunderlich at the Bionics Institute and the University of Melbourne, Australia. So over the past 4 years, I've been working on an exciting project that involves using fNIRS to measure speech discrimination ability in sleeping infants. So due to the COVID-19 pandemic, so I was presented with the opportunity to develop a novel method for extracting heart rate response from fNIRS data, collaborating with my supervisors and the engineering team in the institute. So the details of today's webinar can be found in our published paper, and please feel free to reach out via email if you have any further questions. So let's start with some background information about this study. So listen to this. Ba, ga, If you can hear the syllables that I just said, it means you can detect the speech sounds. If you can tell the two syllables are different from each other, it means that you can discriminate the speech sounds. Now try to imagine if you couldn't differentiate between sounds such as sh and s sounds. Often not, you will find yourself having difficulty, to process and understanding sentences that have the words such as she or she in it. So at this point, you might be wondering, like, why do we even bother, to measure such a simple ability? Like, is that necessary? And don't we develop it, naturally? So indeed, studies have shown that, babies can discriminate various speech sounds. And in fact, they can do better than adults. However, the same thing does not apply to infants with hearing loss who do not have access to good sound quality during the prenatal period due to the limited hearing acuity itself. So they do not get the same opportunity as their normal hearing peers to develop their brains to process the sounds, and up to the level where until they got fitted with a hearing amplification device. So there are quite a big gap between them and the normal hearing peers, and there's a crucial need to close this gap, the performance gap between infants with normal hearing and infants with hearing loss itself. So currently, the most used speech discrimination test is the behavioral speech discrimination assessment called the Visual Reinforcement Infant Speech Discrimination Test, also called a VRSD. So in this test, we get the infant to sit on the parent's lap and to look forward, and then we present them with a standard stimulus, such as ba sounds continuously at the background. So it's going to sound like ba, 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 ba. And then when we present a novel stimulus, such as ga sounds, we will condition the baby to turn and look at the reinforcer, like that, indicating they discriminated the speech sounds that they just heard. So at this point, you might be able to see for this test is not applicable to all babies, especially very young infants who do not have the necessary head motor control to perform the head turn movements. So they're just too young to do that. Also, some of them just could not be conditioned, their brain hasn't developed fully yet to perform such tasks, and they might not have the attentions, to do the whole test itself. So that's why there's a pressing need for an objective test in order to overcome and to help the clinicians, to better identify and better measure the speech discrimination ability in very young infants in order to help them in the pathway of rehabilitation itself. So when there's a change of sensory stimuli, our body actually generates an autonomous response, such as the heart rate changes. So here I'm going to share with you a study from the Schustertians and colleagues. So in her study, they measured the group average heart rate change in a group of adults by presenting them with noise at different intensity levels ranges from 15 to 90 dB SPL. So when the adults were presented with louder stimuli, such as 65 and 90 dB SPL noise, Schustertians reported that the group response showed an increase in heart rate, as you can see in the black and the red line in this figure. So there's an increment in the heart rates when they listened to a louder sounds. However, when they were presented with a moderate or a softer sounds, such as 40 dB and 50 dB SPL sounds, the heart rate actually decelerates. You can see a decrement here in the blue and the green line in response to the softer auditory stimuli itself. So in infants, several studies have demonstrated the use of habituation, dishabituation test paradigm to modulate heart rate response. So I would like to share with you a study by Moffitt, which investigated the use of heart rate responses to measure speech discriminations in awake 6-month-old infants. So Murphy found that the average heart rate response decelerated after the first trial of a standard stimulus of bar sounds. So Murphy presented bar, 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 bar sounds, and they reported a deceleration heart rate response. Following this, he presented another 5 to 6 more blocks of standard stimulus block and reporting a reduction in the response amplitude until it returned to baseline. And this is suggesting there's some habituations happening here with the heart rate responses. Then he presented a single trial of novel sounds, which is the ga sounds, ga, 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 ga. And you can see there's a renewal of deceleration response following the presentations of a new stimulus itself. Hence, you might be thinking, oh, this sounds like what you're doing. So in my study, I aimed to find out whether these test paradigms can be similarly used in sleeping infants and to demonstrate speech discrimination ability through the response to the novelty stimulus itself. I'm also interested in determining whether this this heart rate change is sensitive to the degree of contrasts between different speech contrasts, and also whether the dishabituation response actually, habituates with multiple repeats of the experimental runs. So the reason that I'm interested in this is because we want to develop this tool for clinical use. And for clinical use tool, it has to be able to measure speech discrimination response in individual infants, and hence multiple repetitions is necessary in order to reach a reliable conclusion itself. So we like to know whether the response actually habituates with multiple runs, and that's going to affect how we develop this test as a clinical tool itself. So fNIRS is a neuroimaging technique that measures the blood changes in blood oxygenations using near-infrared light. So the light is emitted through the optodes placed on the scalp, and the light travels through the scalp and to the brain tissue. And the lights that reflected back is measured by the detector. So typically, fNIRS raw data can be decomposed into multiple components, such as the hemodynamic response, which everyone is familiar with, and also the cardiac signals. As shown in the figure of the right side here, you can see here, which is the power spectrum of the raw data of in infants data. So you can see there's a very prominent heart rate signals at 2 Hz and it's harmonic. So usually in a typical fNIRS processing pipeline, we will put a bandpass filter to remove this type of systemic responses. But in this particular study, we are interested in this heart rate cardiac signals, and we wanted to see how it's be able to use to measure speech discriminations in infants itself. So the hemodynamic response in this data set has been published in the paper by Maud et al. And also a few other studies have demonstrated that fNIRS hemodynamic response is sensitive to the habituation-dishabituation paradigm. So ultimate goal of developing this heart rate technique is to enable us to combine both the fNIRS hemodynamic and heart rate changes in order to improve the accuracy of measuring speech discriminations. So we hypothesized that the heart rate changes with the onset of speech stimuli, indicating speech detections. We also expect that the heart rate response changes to novel stimuli will be larger than the standard stimuli, which indicates a speech discrimination ability. We also hypothesized that the magnitude of the discrimination response will be sensitive to the degree of acoustic difference between the standard and the novel stimuli. And finally, we predict that the detection discrimination response will decrease with multiple runs of the experimental paradigm. But we also would like to know whether if we alternate the speech contrast between runs, will it mitigate this adaptation effect and preserve the novelty effect? So in this study, we recruited 26 infants to participate in this study, but 3 infants were excluded. 2 of them did not sleep in the lab, which is pretty normal in a sleeping lab, I guess. And 1 of them failed the middle year check on the test day itself. So the final 23 infants had no known hearing loss, and all parents reported no concern with the infant's health condition. So the stimuli used in this study were natural recordings of consonant vowel syllables, the CD syllables, and the syllables were paired into 3 different speech contrasts, which were Bati, Bubi, and Shisi. So the Bati is expected to be the most discriminable contrast as it contains both vowel and consonant differences. And Shisi is expected to be the least discriminable contrast as it contains only the consonant difference. So the fNIRS data were recorded using the NIRx device, coupled with the flat infant optodes, as you can see on the figure here. And then the montage you can see here was designed based on the Nakano study. So for the purpose of this study, the optode placement is not really relevant because the heart rate information was extracted from all channels and combined. So all participants were tested during natural sleep, held by a parent, sitting on a very comfortable couch in a very dark room during the fNIRS recording process itself. So the stimuli were presented using the habituation, dishabituation test paradigm, adapted from the Nakano study as well. So in each round, 10 blocks, you can see here, 10 blocks of standard stimulus were presented, and they were grouped to a group of 5, to a block of 5, and named as HAP1 and HAP2. We expect that the heart rate change response to reduce in amplitude from HAP1 to HAP2, as shown by previous studies. So we expect that to happen. And then we present 5 blocks of novel stimuli, which is, for example, T, 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 T. And we hypothesize that the heart rate response for the novel condition would be larger than the habituated HAP2 condition itself. So we expect novel here to be higher than HAP2. So what you can see on the speaker here on the screen here is just one run of the stimuli presentations. And so when we run this study, we repeated multiple runs until the baby wake up itself. So we have unequal number of runs per babies, but that's the conditions of testing sleeping studies in general, because some babies sleep for 30 minutes, some sleep for 50 minutes, some sleep more than that. It's just hard to predict what's going to happen in a sleeping baby clip. So next, I will walk you through the data pre-processing and the heart rate extraction method itself. So the data pre-processing was done using the brain analyzer toolbox by the Hubbard's group and also some custom script that we developed here. So we first evaluated the signal quality by using the scuff coupling index. We found a total of 7.48% of channels to have scuff coupling index threshold below 0.8, and they were discarded. Then we converted the data to optical density, and we bandpass filter the data between 1.5 to 3.33 Hertz. And this filter range corresponding to 90 to 200 beats per minute, which is the average range of heart rates for infants below 2 years old. And then lastly, we normalize the data in each channel by dividing the data by its envelope itself. So now I'm going to go to the exciting part of this study, which is the heart rate extractions methods from the fNIRS data itself. So in this study, 8 sources were switched on sequentially because we selected the sequential illumination patterns in the NIRs software configuration tab itself. So as you can see in this slow motion illumination pattern, source 1 lights up first, followed by source 2, 3, 4, and then till 8. So even though the data points from one source are 0.128 seconds delay compared to the previous source, all data points from one illumination sequence is actually stored onto a single timestamp as highlighted on the red bar that you can see here. So even though there were slightly delayed between them, there's a slight delay between them, but they were stored onto a single timestamp in the NIRx machine itself. So what we try to do here is that by applying the correct time offsets for each source according to the illumination pattern, the effective sampling rate. So this is the figure left is the same figure as the previous figures. So when we apply the correct offsets, and it results in the figures on the right. And by doing that, we actually able to restore the sampling rate from 7.81 samples per second, you can see here, to 62.5 samples per second. So we effectively increases the sampling rates without using any upsampling that involves interpolations between data points itself. Then we average the multiple data points stored for each timestamp. So you can see some timestamp have multiple data points because they're connected to the same source. And then we smooth the waveforms using a 10-point moving average. And we smooth that, and you can see depicted by the black line figure here. So to extract the instantaneous heart rate from the waveform itself, we have to first identify the zero crossings of the cardiac waveforms, where the data changes from negative to positive. And then we calculated the interpeak interval by calculating the difference between two consecutive zero crossings. And lastly, we calculated the heart rates and yielded the instantaneous heart rate, which you can see in this figure. So this is an example of the process heart rate versus time waveform for one run, only one run, from one participant itself. So that's all. That's pretty much all for the methods of this study. So moving on, I'm gonna share with you the results of what we found of running this exciting study. So, sorry, before I move on, I will talk to you how I extract the response size first, so that you know what I'm doing. So the data from the instantaneous heart rate that we extracted from the fNIRS data, we epoch them between -3 seconds pre-stimulus onset and also 14-second post-stimulus onset. And then we converted it to a heart rate percentage change from the baseline itself. So to extract the response size for each run of the stimuli presentations, we will first average the epoch waveforms across all the different conditions, HAP 1, HAP 2, and normal, within the run itself. Then we will extract the response size, which is defined as the average values within a defined time window. So in this study, we define a time window by marking the time before and after, where the grand average amplitude fell to half of the peak amplitude itself. You can see here is half of the grand average peak amplitude, and the times that marks the intersections here is the time window that we use for this study. And the time window that we use is 1 point. I think I got it on the next slide. I'll talk to you in a moment. After that, we repeat steps 1 and 2 for other runs. So as I mentioned earlier, this is just one run, and every baby has multiple runs, and we do that for other runs as well. So this is a time window that I mentioned just now, which is 1.75 to 6.38, and we average the values within them to extract the response size. So the figures that you can see here is the group average seamless evoked change in heart rate, which is averaged across all the conditions, all the runs, and all participants. So this is the grand average speech detection amplitude itself. So we also did, we extracted the speech detection's response size using the process I just described in the previous slide. And we run a t-test against 0, and it shows a significant acceleratory response, a significant increment of response compared to 0, which shows that the heart rate accelerates with stimuli, speech stimuli, in response to speech stimuli. So in this figure, we're going to show you demonstrate how the heart rate change response habituates over multiple experimental runs. So in this study, 6 participants actually listened to repeating experimental runs of the same speech contrast. So if they got bar T speech contrast, for example, they just listened to bar T throughout the whole experiments. If they got booby, they just listen to booby, and if they got she-C, they just listen to she-C speech contrast. However, 14 participants actually listened to runs that alternated between the 2 speech contrast pairs. So for example, if they were started with bar T, then the next one could be she-C, and then it just keep alternating bar T and then she-C again, and then bar T and then she-C again. So 14 participants actually listen to alternating stimuli presentation paradigm. So in this figure, there's a clear decreasing trend of the speech detection response size over multiple runs. And this result is supported by linear mixed model analysis, which shows a significant effect of runs, but no significant effect between the 2 different conditions or protocols. And also there's no significant interactions as well. And then we did a post hoc analysis, and it shows that 1 is significantly different from 2, and 1 is significantly different from 3, but there's no significant difference between 2 and 3 itself. So now I will share with you the results of the speech discriminations analysis. So previously we're looking at speech detections where we just average across the whole condition itself. So now we're going to look at speech discriminations. So one more time, for each experimental run, we average the epoch waveforms per conditions. So we average across the epoch waveform for the 5 stimulus blocks in each condition itself. Then we extract the response size for each conditions using the same methods described in the speech detection just now. And then we compute the discrimination response size by subtracting the response size of HAP2 from normal. So we use the response size from normal and minus the response size for HAP2. And then repeat steps 1, 2, 3 for all other runs. So this figure here is the group average heart rate change response per conditions, which has been averaged across all contrasts and participants. So the first thing that we can see here, very clear, is that there is stimuli evoke responses for all 3 different conditions. No matter HAP1, HAP2, or novel, we still do get stimuli evoke response. However, the yellow line here, which is the novel condition, had the highest average peak amplitude, whereas the HAP2, which is the red line here, had the lowest average peak amplitude. We also did stats using the speech discrimination response size, and we have shown that it's significantly greater than 0 as well. So we introduced different speech contrasts in order to investigate the effect of different discrimination cues, on infant heart rate change response. So from this figure, we can see a clear discrimination response for Bati and She-See speech contrast, but not so much for Ruby speech contrast itself. But when we did a linear mixed model analysis, it shows that there's no significant effect of contrast pair on the discrimination response size and no significant interaction as well. And we think that this lack of effect could be due to all speech contrasts being, discriminable for all the participants. It means that they are all pretty easy for the baby. So that's why that could be a reason why there's no difference in the heart rate change response between the different speech contrasts itself. And then the last results I would like to share with you is to investigate whether there's a habituation of speech discrimination's response size with repeated runs. So you can see here, there seems to be a trend of decreasing response size over the multiple runs. But the stats show there was we did not find any significant statistical findings itself. But from the visual observations, there seems to be a reduction in the response amplitude as well. So moving on is the discussions. So our results have shown that the heart rate response can be evoked by the detection and discrimination of speech token stimuli, similar to the previous findings in awake babies. But in contrast to the deceleration heart rate response reported in previous studies, we found an accelerating heart rate response to speech in response to speech stimuli. So this major difference we think could be attributed to the testing states, where the previous study measured response from awake infants, and we measured the response from sleeping infants itself. So the mechanism of this acceleratory heart rate response remains unclear. But one of the study by Anderson's reported that the accelerating heart rate change response during sleep is suggested to be a defensive reflex. So this defensive reflex is potentially enables an infant to maintain their sleep state while staying alert to the environment. So even though the mechanism remains unclear, it is still a useful biomarker for speech detection and discriminations. So this heart rate response can be available diagnostic tools that add value to the test battery for measuring speech detection and discriminations itself. This potential tool could be useful for assisting in measurement of hearing ability in individuals that could not be measured using behavioral tests, such as infants. And it is definitely an absolute advantage that this response can be recorded in a sleeping state, because a lot of previous heart rate change response in awake studies, they have to discard the rejection rate is almost 50% of subjects due to the difficulty to get the baby in a calm and quiet state. So just imagine you ask a baby to sit down and look forward and just don't move, and they don't listen to you. So in this study, we used the fNIRS recorded heart rate response, and it is an additional information from the fNIRS raw data that could be used to supplement the fNIRS hemodynamic response analysis. Since the heart rate response adapts rapidly with time, it might not be a suitable standalone measurement of individual speech discrimination ability that requires multiple repetitions to achieve reliable conclusions. However, the heart rate feature can be proved to be useful when combined with traditional fNIRS features to improve the accuracy of measuring, for example, speech detections ability itself. So the conventional heart rate measurements use technologies such as ECG or pulse oximeter. And the main drawback of fNIRS is its lower sampling rate. So at conventionals, pulse oximeter can go up to usually around 100 hertz, 50 hertz to 100 hertz sampling rates. And the fNIRS sampling rate is lowered when the sampling rate of the machine is divided by the channel that you have. So the more channel you have, the lower sampling rate that you have. But we have successfully overcame that limitations by recombining samples from all the channels and restoring the sampling rate to the maximal sampling rate of the device itself. So in conclusions, we have demonstrated that speech detections and discrimination ability can be measured at a group level using the heart rate change response in the fNIRS raw data. So future research is needed to devise testing protocols that limit the adaptations of the novelty response over multiple runs in order to improve the reliability for testing individual infants itself. So here I would like to acknowledge University of Melbourne for providing me with a scholarship to do my PhD. The Bionics Institute, who has been a great support through my 4 years journey here. Victoria Infant Hearing Screening Program, who helped us with the participant recruitment. And last but not least, the Human Hearing and the Ear-Genie team in the Bionics Institute for enabling this research project itself. So these are my references, and thank you. That's all for my sharing today.
Thank you very much, Onn Wah, for that really great talk, and congrats on the awesome work. So the floor is open for questions. If anybody in the attendees has any questions, you could type it into the chat, or you could use the raise hand function and you can directly ask your question. In the meantime, we'll get us started with a few questions that came in during the registration. So there is a question about heart rate variability. So the question says, are you looking at heart rate variability at all?
Yeah, so heart rate variability is a very big field. When I first started, I decided, when I first started, I did look at heart rate variability, the term itself, but in the end, we decided not to use that term and we use heart rate change response instead. Because I've seen that a lot of heart rate variability study involves like very long hours. They actually look at the changes, startle changes over a very long durations. But in general, by principle, what we are looking at is heart rate changes within time itself. So it is a variations of heart rate within a certain period of time. But it's just that the time that we're looking at is very small period, which is just around 14 seconds post-stimulus onset.
Okay, perfect. And another question, again, this one came during the registration. So you did expand a little bit in your data analysis, but the question is, can you offer some more details on data processing and the analysis? So besides what you've mentioned in your talk, is there anything else you would like to add to that?
Yeah, actually, I simplified the signal processing here, not to bombard Aaron with too much information. So it's not as simple as I've shown here. So there's a need for signal cleaning. There's the need to clean the signals, which involves rejections of noisy section of the data, which I did not include in these presentations, but it's available in the paper. So if anyone's interested, can refer to the paper where I describe in details on how I reject noisy data itself. Also, just on a quick note, while we're talking about signal processing analysis here, this method also applies on adult data. So I've tried it. I mean, we did not publish any results on that, but we tried internally, and it works pretty well as well.
Awesome, it's perfect. Yeah, we encourage everybody who's interested in the analysis to definitely check out the publication. And one last question about analysis as well. It's asking about, is it possible to see the Monte Carlo sensitivity 3D maps? So something like what you would get from Atlas Viewer.
Yeah, so I saw that questions and I added it on my slide here. So this is the 3D heat map of the montage. But however, I need to emphasize that as I mentioned in my presentations, we actually combine the data from all channels. So this atlas viewer actually doesn't really mean much because in fNIRS, I know we focus a lot on region of interest, and measuring from different locations. But heart rate is a systemic response; it's a global response. So we make an assumption that it's the same heart rate that appears within one sequential illumination pattern. So, yeah.
Perfect. And I see there's a question from the attendee. So Julia saying, thank you, this is amazing work. If I understand correctly, during the fNIRS recording, the light sources were turned on sequentially. Is this a necessary step to extract heart rate?
Oh, so it's a default. So default is the standard illumination pattern is a sequential illumination pattern because that's how fNIRS equipment works in general. But there's an options of doing bilateral illumination pattern in the NIRx NIRscope device. And if you using that options, you just have to map the offset correctly to each source itself, and that will be fine. Yeah. So it's a standard to do sequential illumination pattern.
Perfect, thank you. I see, Eric, do you have any questions?
Yeah. Thanks so much for the talk. If I understand correctly, this is probably the same data from your 2021 paper. Is that correct? So you're looking at infants from 2 to 10 months of age. So there could be some maturation potential effects, I guess, with respect to like an evoked hemodynamic response. Is that something that you've looked into? And then also, does that maybe exist within the heart rate data that you're looking at now?
Oh, great. That's a great question. Thank you, Eric. So so we all know there's exponential development of the brains in their first year of life. So for this paper in 2021 itself, we did not look at the changes with age, but actually, I mean, like we did the analysis within the institute itself, but the results hasn't shown any, hasn't seen any trend using just the 16 subjects that we have at that time. So we are still waiting for. Actually, no, I wouldn't say we're still waiting for. So there's some other studies using the same data set that look at functional connectivity, and we do see some changes between the very young and the very old group, which is like 13 months old and above. But generally, there's not a significant change in the data that we recorded so far. Also, because speech discrimination is a skill that develops at birth. So in these normal hearing data sets, we do not really expect a developmental effect on the speech discrimination response. But we're definitely interested to look at the developmental effect on the fNIRS response itself.
Okay, great, thank you. Did you also anticipate any differential effects within, so this is of course when the infants are sleeping, would you expect anything different? You said there was maybe a compensatory mechanism, you said to, I guess, preserve sleeping, but how might this differ to an awake subject?
How does this different to the awake subject? So, the reason we suggest that is because in the visual visual habituation fields in infants, it's a pretty developed concept that the heart rate change actually decelerates, as you can see on the figures here. And that is the like a defined, the visual fields uses to define attention spans in infants itself. So when the baby pays attention, the heart rate decelerates. So in sleeping infants, first of all, we do not expect the baby to pay attention, because paying attention is a conscious act, and you are sleeping, so you're definitely not paying attention. So so we definitely do not expect. We are not surprised that it's different. Also, secondary is also because generally you are sleeping and your heart rate is only get triggered when it's necessary. So you do not want, you can't have a very fluctuating heart rate. In other words, when you sleep, because that's not healthy to have a good sleep state. And that's why that's why the mechanisms that we think most likely is the defensive reflex that I mentioned just now, where because an increment of heart rate is the preparation for fight or flight response. So when you're in a tense or when you're in threat, your heart rate actually shoots up, increases. So and that's a defense mechanisms, and that's why we think that is more likely to be the case than paying attention. Yeah.
If I may add just very quickly to build up on Eric's question, would you expect to see any differences with stages of sleep? So, deep sleep versus more like light sleep, REM sleep, in terms of heart rate, in terms of the responses in general?
So that's a very interesting question. We had that in our mind as well, because studies have shown, the heart rate changes varies with sleep state a little bit, varies with sleep state in general. However, we are not looking into that. First of all, because we are evoking a response, so we actually, actively presenting stimulus. So it's not like a steady sleep state. It's not resting state. It's not sleep state. Sorry. So we are actively introducing sounds, and it's an active response that we're looking at. But definitely, it's sort of come across my mind as well. To actually, in future I might just want to, pop two electrodes on the baby's head and just simultaneously record the sleep state. But in general, in babies, everything is different. With adults, in babies, even the sleep state is different in baby. The baby is like 50% of REM and non-REM sleep. So so generally everything is different. So it's a bit hard to use anything to make inferences based on studies that done on adults itself. So yeah, but it's definitely within the interest to just stick to electrodes and just get the EEG and identify the sleep state itself.
Perfect. Sounds good. Are there any more questions? I'll just take a quick look at the chat. Okay, I don't see any more questions, but of course, if anybody has any questions and you want to ask it afterwards, please email us consulting@nirx.net. We can forward your questions to Onn Wah directly, or you could also email him, of course, directly, and you can answer them that way. Again, this webinar is recorded and will be made available to all the attendees and will be shared with everybody that registered as well. Thank you very much again, Onn Wah. This was extremely informative and a really great talk, and we're very much looking forward to all your future work as well. Thanks very much for joining us.
Great. Thank you. Thank you for having me. Thank you for coming, everyone, especially from Europe. I think it's midnight there.
Yeah. Awesome. Have a good one. Take care.
All right.